Main issues as far as the technology is concerned, you may have noticed right at the beginning, I'm recording these lectures. They're going to be on YouTube. So that's all there. Again, that's the main reason I'm worrying about it. Just to get started, a little bit about me, where you can find me. I'm over in the building previously known as SB2. For those of you who've been here around long enough, that's SRTC, up on the 4th floor on the west side of the building. I have office hours right after class. We'll get to that in just a second. Some of you may have seen that picture over on the right-hand side there. I chase viruses sort of for a living. So the virus hunter, that's what the work is going on in my lab. I'm working on a number of different things, including a movie called Edge of Life, which some of you have probably been subjected to already, at least the trailer thereof. And most recently, I had my arm twisted into starting a company that we can talk about later, too, called Stone Stable, about vaccines. What are we talking about for the next 10 weeks? Basically, sort of the fundamentals of molecular biology. Start out with a little bit of chemistry and genetics in terms of review. If you haven't had these kinds of things, they're really new to you, we can talk more about that a little bit later. The main part is really about nucleic acids as far as this course is concerned. DNA, RNA, structure, and certainly our genomes. Talk about how you make more DNA, how you repair DNA, particularly talk about how DNA is actually not a really great molecule for storing genetic information, but it's what we got, so therefore we're interested in it. How you go from DNA to RNA, and then from RNA into proteins, translation, and then we're going to spend a really long time talking about regulation, because that's really probably most important about diseases, development, sort of you name it, in terms of how our genetic information becomes us. So it's really a lot about the regulation. And then RNAs, and RNAs in particular, in terms of how they're functioning, particularly from a regulatory point of view, how RNAs are regulatory RNAs. And this is something which is really, probably in the last 10 years or so, people have finally realized how important RNAs are for regulatory purposes. What do I expect you know? Basically, everyone I hope has had Gen Chem or something equivalent. Everybody, yes? Gen Chem, good. Principles of biology, at least the first term. The whole term, the 3-course segment is probably not as critical, but certainly the first term. A lot of the stuff we're going to be doing in this class is following up on a lot of that. How many people have had genetics? Good. That will help, not critical. OChem. Okay, we're going to cover basically everything you need to know about organic chemistry on Wednesday. Biochemistry. Okay, that'll all be covered in about 5 minutes on Wednesday. Microbiology. Yes. Microbiology is totally cool. Not quite as cool as virology, but close. Cell biology. Okay, in theory, this is a prerequisite for cell biology. So I know some people end up taking them out of order. That's fine. Course materials. The main thing about this class is the lectures. Yes, we've got textbooks and so on and so forth, but everybody asks me, where do you get your exam questions from? And the answer to that is, I get them from my lectures. I don't go back to the textbook. I don't go and Google something really obtuse online and then put it on the exam. It really comes from my lectures. And that's partly why I put my lectures online as well, just so you have access to them. That being said, the lectures are based to a great extent on the molecular biology of the cell. This is the 6th edition, which finally came out in December of this year. It's actually the problems book, but the rest of the book looks pretty much the same. The main reason that we've gone to this text is A, it's up to date, and B, it's probably sort of the classic molecular biology / cell biology textbook. So we use the same textbook for both. I know the textbook is ridiculously expensive. That's unfortunately part of what we have to live with these days. We're trying to deal with that as best as we can. Part of that is to try and use the same textbook for 2 courses. There actually probably are better textbooks for molecular biology. I don't think there are any better textbooks for cell biology that are out there. We're going to be covering in this course chapters 1 through 8, mostly skimming over chapters 2 and 3, because that should be review. again, based on your Gen Chem and Principles of Biology, which you should have already had. So that's basically going to be these first 2 lectures. Website, D2L, we'll take a look at that in just a second. That's got the syllabus, additional materials, all the PowerPoints. Anybody download today's PowerPoint beforehand? Yes, it was there as of last night. I try and get them up there by 10 o'clock the night before lecture. Also, all of the previous year's materials are there. So all of my PowerPoints for pretty much the last 10 years or so. Unfortunately, as I mentioned with the textbook, stuff changes really fast. So they're not all going to be completely up to date. And then this is the link to my YouTube channel. Again, that's where I'll be posting all the lectures. It's not terribly well organized. I'm going to try and get that better organized as far as the term goes through. But I'll let you know as that moves through. So let's take a quick look at the website here. If we can get this up and going. Hopefully it will. If not, it's not a big deal. Come on. Everybody know about D2L? Anybody not know D2L? Not used D2L before? Okay, good. So it's pretty straightforward. There's some things about D2L that I really like. There's some things about D2L that I really don't like at all. But the main thing is just a place for me to post stuff, including your grades. No, I don't need this. Cancel. No, I don't want my login. So, yeah. So yeah, these look slightly different, of course, than what you see because this is mine, not all of yours. But what I wanted to look at here really briefly is the syllabus, just to go over that. It's here. Hopefully, again, most of you have seen this already. Basically, again, what we're going to be talking about, what the readings are about. Some of you may have noticed that a couple of these are in italics. That means there'll be a guest lecture. I won't be here on this Friday, so we'll have one of my graduate students will be talking then. And then on, I guess this one's not been updated yet, on the 27th of February, we'll also have a guest lecture for that. I've tried to break down more or less what pages we're going to be covering in any given lecture. But again, this most important is really what's in the lecture, not necessarily what's in the quote-unquote reading for that particular aspect of things. Also has the dates, etc., for midterms and final. That's where you can find me, prerequisites, textbook. We'll talk about the clicker in just a second. D2L, again, this is where all of the materials are. 2 midterms, 50 questions each, multiple choice, Scantrons, the new big forms. I'll remind you again about that when that gets a little bit closer. Finals not cumulative, examination policy, grading, etc. Probably more important is down here at the bottom. This is what I hope you're going to know by the end of this class. So this might also be a really nice outline in terms of thinking about studying for exams. These are the kinds of things that I hope you to know. And then probably the most important is really the last thing down at the bottom here, and that's when it was last updated. This is an evolving syllabus, so I will keep changing things on here. I won't change exam dates or anything like that, but in terms of what subjects are being covered, we may end up going a little faster, a little slower in any given section. That will all be there. Any questions about the syllabus while I'm on it? Okay, other quick things as far as course content is concerned. Again, here are the lectures. I'll post them as soon as they're done. This has Watson and Crick's paper from April 1953 on the structure of DNA, which I expect you to have read by Friday if you haven't done so already. Lots more information on DNA, videos, etc. Probably this will be also something you'll want to take a look at. Old exams. All my old exams, all my old keys, they're all posted. online there. So in terms of what kinds of questions are likely to be on exams, that's a really good place to go and take a look at them. And then all previous year's materials. Did want to mention something here really quickly. Fall 2012 is the last time I used this textbook, only it was a previous version of the textbook. So we'll get back to that in just a second. But those are probably better notes for taking a look at rather than last year's notes because we're using a different text. And so it's not going to be quite as comparable between the two of those. And the same thing is true of the lectures, which are also, my lectures from last year are all online as well. That's all I wanted to mention as far as D2L is concerned. There are lots of other resources that are available. Again, last time I taught this course, we used molecular cell biology from Lodish. Again, probably a better molecular biology textbook. I'll be using some of the figures from there in terms of my lecture notes. Our previous text here, Molecular Biology of the Cell, the 5th edition, unfortunately in 2008, and 2008, 2015, 7 years, is more or less a generation as far as molecular biology is concerned. Unfortunately, this field moves really, really fast. Molecular Biology of the Gene, I think they're actually up to the 7th edition now, et cetera, et cetera. Did want to mention NCBI. NCBI Books is the National Center for Biotechnology Information. It's run through the National Institutes of Health. Has books, including, admittedly, only the 4th edition of this textbook, completely available online for free. So if you're really in a pinch, this is a place you can go and get some of that stuff. And these are a couple more textbooks which are there. Unfortunately, that's what we have to deal with. How do you get a grade in this class? Has to do with 2 midterms and a final, each of which are worth exactly the same amount. They're all 50 multiple choice questions each, 28th of January and the 20th of February. The final is just the normal final schedule. It's at 8 a.m., not at 9 a.m., and it's on a Tuesday, not cumulative, and because you see all the TA support that I have here, they're unfortunately all going to be multiple choice, and we'll use the larger versions of that for the Scantrons. But again, I'll remind you about that again once we're there. I will be using clickers. You may have noticed that that was just 30% each. 3 exams at 30% each come up to 90%. So now I've got 10% left, which are for your clickers. The clickers are mostly for me rather than for you. I know it seems unfair that I should be giving you 10% of your grade when, mostly it's for me. So trying for me to get an idea where you're at in terms of the understanding so we can tailor, or so I should be able to tailor my lecture in terms of what we know about. They'll be starting officially in week 2 in terms of counting. I'll have clicker questions this week just to get ready for things, and that brings us to our first clicker questions. So if you would go ahead and get yours out, partly also to make sure the system works before it actually starts counting for people.
So are you just going to do credit for participation, or is it?
So the question here at the front was credit for participation or credit for getting them right. No, you do need to get them right in order to get those questions. So we'll start our session here, if it'll actually do so. It should be the standard one, your AA on here. So let's give it a try, see if it works. Well, at least 30, 40 of you are fine, good. 136 people registered for the class so far. In theory, we can have 150, but in theory, we can also have 200 people in this room, which I don't think we should have. Any more of you have your clickers? You don't want to admit? Yeah. Got it? Pardon? Not everyone apparently has a clicker yet. No, we only got 60 people who are here who have clickers. And there are a lot more than 60 people who are here. You only actually need them the second week. A lot of people ask me, can we use the mobile version, the Clicker Go? No, unfortunately not, for a couple of different reasons. The main one is I don't want you flipping through your phone looking for answers at the same time as you're trying to vote. I think that's a bad idea. And the second one is that so everyone's sort of in the same boat as far as it's concerned. Okay, anyone else want to vote? I usually give people 90 seconds for this, so we'll have that taken care of. Okay, good. And if there are questions that come up with clickers, one of the nice things is these clickers will just basically download everything into a spreadsheet, which has the clicker ID connected to your response. And then basically what needs to happen is you need to connect that clicker ID to your name. And that's what this next one is supposed to be here. Here we are. So those of you who haven't registered your clickers in terms of a name and this class, etc., please do so at iClicker.com by the 28th of January. Need your name, clicker number, and student ID. Something just changed this year. In fact, I got an email about this about 3 weeks ago. Unfortunately, if you have a used clicker that you bought from somebody else, you have to pay to get it registered. I think this is absolutely crazy, but unfortunately, that's the position that we're in. $7 is not ridiculous, but still, it's kind of a drag. That should be a one-time registration, however. If you've gotten a used clicker and registered it through iClicker before, it should still be okay. And I don't know if that many people have tried that yet. Yes, you had a question? Should be the same registration as last term, yes. Pardon? Yeah. I'll have to see. We can check on that. I will post, by the way, all of the clicker scores at the same time as the midterm. And so there'll be a way that you can check and make sure that you're at least getting those scores taken care of. I can't do that right at the beginning. Again, if there are 130 of you, I can't go through and check each and every single one of them. But by the time the first midterm comes along, I also have a pretty good idea in terms of the number of people who've been voting to get an idea of how many people we actually have and have been appropriately registered. I will come and try and find you, because there are usually 5 or 10 who aren't registered at some point. Other questions about clickers at this point? Okay, how do I do my grading? It's pretty straightforward. The top score is set to 100%. And that basically means that I make a couple of bad questions on exams and people miss them. And what I do to normalize that is say that the top score is set to 100%. 90% of that's an A, and so on and so forth. Makes life a lot more straightforward so you know where you are. And I will post what those top scores are on D2L, so you can immediately go through and check and see where you are. I'll also be keeping track of the clicker scores, and I'll post those as well. How do you do well in my class? Study old exams on D2L. I wouldn't want people to force people to listen to me more than once in any given lecture, but apparently it works well. YouTube channel, office hours, which are right after class. How many people have a class right after this one? Okay, so quite a few of you. I'm here basically from about 8 to about 5 Monday through Friday, so we can set up some times to meet outside of when I'm here, of course, and that other lecture time. I highly recommend study groups. You can use D2L, hopefully, to get through to study groups. And there's what, My Buddy, is it? What's the? Buddy Up. I haven't used it, but I've heard that some students find it to be very useful. I will have a review session before the exams. In fact, the last lecture before any exam is just going to be a recap of what we've covered before then. Maybe outside of class, we'll have to see if we can organize that. And then, as I've heard from many students, I talk too fast, go over too much stuff. The best way to slow me down is to ask questions. And if you have a question, it's highly unlikely that you're the only person who has that question, particularly if there are 136 of you. Okay, I have to tell you about cheating in attendance. I don't require attendance, but of course your clicker scores might be thought that way. That being said, I do still highly recommend that, and the reason for that is I've actually done some surveys. I have some data. It's from a couple of years ago, even before clickers. Almost always come to class. Blues are A's, reds are B's, whites are other things, most of the time, sometimes, rarely. You can get an A if you come rarely, but the people who usually end up with A's are the ones who come much more often. Makeup exams, extreme circumstances, et cetera, et cetera. If you go back and look at the notes for February of 2012, you may notice that Lecture 1 and Lecture 2 are basically identical. I was in the ER for Lecture 1, so that's why they're slightly there, and I thought that was a decent excuse. Syllabus, again, we looked at this already. Communication, I think, is really important in terms of getting the material. Again, part of that's this clicker. Discussions, I do follow all of the discussions on D2L. So if you want to discuss about me, maybe that's not a good place to discuss it. But I will first try and let the rest of the people in the class make comments in terms of questions, but then I will certainly jump in at some point, looked at least once a day. If you have a specific question that you think might not be more general or something more about the class rather than about the subject matter, feel free to send me email. I'm kind of email dependent. We'll be reading it more or less constantly. If I think it's something which is really important for the rest of the class in terms of subject matter, I'll post that to D2L, but I'll take your name out of it. So if you want to have something posted anonymously, I can certainly do it that way. Okay, any questions about the class in general before we move into some of the subject matter? No? Okay, good. Otherwise, again, send me an email, post things to the discussion group. One of the things on the discussion was about using the 5th edition of the textbook versus the 6th. It's there. You can take a look at it. So today, I wanted to talk about the 3 domains. So everybody in microbiology already knows this, but I think it's a really important concept to think about. And then the central dogma, which is that DNA goes to RNA, goes to protein. And then we'll talk a little bit about these different, again, this is very much a review in terms of what we'll be covering in more detail later on. And then a little bit about genomics towards the end, which should be about 10 to 10:05. Start out talking about the 3 domains. Hopefully this is something which is not new to any of you. If you think about how all life is related to each other, and when I say all life here, I mean all not viral life. We can argue about whether viruses are alive. It's a totally different question. But at least if you compare molecularly all organisms to each other, all cellular organisms, they're all related to each other. And they're related based on their really fundamental molecular aspects. And one of the most fundamental things we know about cellular life is it uses ribosomes to make proteins. And those ribosomes are the same in terms of their structure and also very similar in terms of their nucleic acid sequence in all cellular life that we know of. And by just comparing these sequences to each other, we can get an idea how different cellular organisms are related to each other. So the classic version of this is what's shown up here in the upper right-hand side. This is now taking the sequence of the small subunit RNA of the ribosome. There are 3 really big groups of organisms. There's bacteria, when most people talk about E. coli. There's eukarya. You are here. This tiny little insignificant twig in terms of molecular biology. And then archaea, which are the group that my lab particularly works with. This here is shown as a tree in such a way. This was originally brought up by Darwin. The first evolutionary tree probably was drawn by Darwin, his Origin of Species, where this junction here represents the last common cellular ancestor of all known cellular life. Now, these are all drawn as nice straight branches. It's probably much more of this blob that we have down here. That's what's from your textbook. And again here, Archaea nicely in the middle because these are the most important ones, and Sulfolobus, the organism we work with, right in the middle here. But then eukaryotes, and again, what I want to emphasize here is the length of these lines represents the difference in terms of sequences between each of these given organisms. And you can see that humans, maize, which represents all plants, and yeast are all extremely similar to each other at a molecular level. So just studying one tells us a lot about the other, again, at a molecular level. And then if you want to learn something about how things work in a really general case, you should be studying things like the archaea, i.e. our Sulfolobus, or the bacteria, over here, E. coli. Now, the other thing to mention here is that all bacteria are not E. coli, all archaea are not Sulfolobus, and certainly all eukaryotes are not humans. So there's also a considerable amount of differences which are there as well. And that's emphasized here. Everything starts out, and these are all eukaryotic cells here, but a lot of bacteria look pretty similar, at least the coccoidal bacteria and archaea as well. Look pretty similar to each other, but they end up looking really different relative to each other down here at the end. This is your sea urchin, your mouse, and your plant cells here. But again, they all look very, very similar cellularly. In terms of the molecular level, they're also extremely similar to each other. What does it mean? It means that, okay, yes, they look the same, but how are they different relative to each other? That difference really comes from the DNA, and the famous example of this is, of course, Dolly here, the cloned sheep. You can take the DNA from an adult organism, put it into an enucleated egg, and end up with something with that genetic makeup of not the surrogate mother, but the actual DNA donor mother in this particular case. This was actually shown considerably earlier for frogs, etc., but this is sort of the famous case right here. We'll talk a little bit more about this, or I should say my graduate student will talk more about this, is sort of the way that people finally agreed that DNA was the genetic information was in the study of viruses and looking at viral DNA, and it was that viral DNA which is being inherited as opposed to the proteins which were thought, in fact, at the time to be what contained the genetic material, what got passed along from generation to generation. So again, it's all about the DNA, DNA to DNA, DNA replication. DNA gets copied into RNA, which is transcription. RNA gets made into protein, translation, and this has to be regulated in some particular way. This is basically the whole course on this slide. And of course, it's all the details on how all of this actually happens in terms of this process. But when I say the central dogma, that's a term coined by Francis Crick, or at least attributed to Francis Crick, and that is that DNA goes to RNA, goes to protein. And that's, again, all you need to know about molecular biology. But it's biology, so there are always exceptions. And of course, the really obvious exception coming from the virus world is that RNA can go to DNA. What's that called? Reverse transcription, exactly. And people thought that was just viruses. It turns out there's lots of cellular reverse transcriptases as well to do the same job. So RNA can go to DNA. DNA serves as a template for making more DNA. And there even are some examples of protein leading to changes in protein. So and we'll talk about the prions about that a little bit later on in the class. And so this big arrow here over here is supposed to represent all of those regulatory mechanisms, or the environment, which leads to all these DNAs, which are more or less the same, turning into organisms which are clearly very, very different relative to each other. So DNA, our friend, the genetic material for all cellular life. There are a couple of things that I wanted to emphasize about DNA. One, that it's polar Polar is not a really good term. Unfortunately, polar gets overused in terms of molecular biology and chemistry. Here we're talking about polarity in a molecular biology sense, and all it means is there's a start and there's a stop in terms of polarity. And so if you think about DNA, one end is not the same as the other end, and of course that's the 5' end and the 3' end. You're going to hear me talking about 5' and 3' a whole bunch in this particular class. One of the really interesting aspects about most DNA is it's not only polar, it's got a front end and a back end, but the front end and back end always like to lie down opposite each other. And so it's a symmetric molecule. Again, most of the time your DNA is double-stranded. So you've got a 5' end over here, which matches to a 3' end of another piece of DNA. That 3' end of the first piece of DNA is going to match the 5' end over at the other end of the DNA. Of course, complementary. This should all be review to most of you, hopefully. You've got C's, A's, T's, and G's. Each of them are going to pair with each other. So anytime you've got a guanine residue, that's going to pair with a cytosine residue, adenine with thymine, etc. So that gives you this process. Here's your single-stranded DNA. You add individual nucleotides based on what their complementary nucleotide is on the other side. Of course, that always brings up, chicken and egg question. Okay, you're always getting complementary. How do you start? We'll talk about that a lot more later on. And this process is replication. And again, for those of you who have seen or unfortunately have had me for another class before, anytime I've got something in red, that probably means it's a really important thing to remember. So this whole process is replication. So going from DNA to DNA, as far as we're concerned for this course, is replication. So it's DNA to DNA. Another thing I wanted to mention, and we'll get back and talk about this more later, if I could spell it properly, that would be redundancy, is that DNA, as I mentioned before, is probably not the greatest in terms of genetic material. It's really long, it's not very efficient, it falls apart all the time, but it has one really great redeeming feature, and that is you've got a backup copy all the time. So the information that you need for DNA is on one strand, and because the DNA is complementary, you have this redundant copy of that genetic information. And as we'll see when we talk about DNA repair, that's really, really critical in terms of knowing, they're anthropomorphizing here, but for that DNA to be able to figure out what the problem is and how to fix it. And so again, that redundancy is really critical. What that also means is if you have a problem on both strands simultaneously, it can be a really big problem because you don't have that actual backup copy to take a look at. RNA, of course, pretty similar to DNA. What's the big difference between RNA and DNA? Not so much that they're single-stranded per se. What's DNA, RNA? One letter, right? Deoxyribo and ribo. So that's probably the major difference, certainly in terms of you look at a whole strand. It's that ribose versus the deoxyribose. But also, of course, uracil, which is being used instead of thymidine. But here, more importantly for molecular biology, certainly at this very overview level, is that when you make RNA from DNA, you usually make many, many copies of RNA based on the DNA that you start with. And so RNA is, yes, a messenger for moving the information from the DNA towards proteins, or actually that RNA being used just by itself. But really importantly, and unfortunately I think it doesn't get emphasized enough, is it's an amplification step. You can make many, many copies of one piece of DNA really easily with RNA. And that's the process of transcription that we'll talk about much more later. Yeah?
Do you mean that the different pieces, are they the separate proteins, separate messenger proteins, or are they just the whole script, multiple copies?
So I'm not sure I quite understand your questions here. So, yeah, so we'll talk about this much more later on. But so the question is, is it, I'll paraphrase here, but is the whole genome that's getting made into RNA, or is it just little pieces of the genome? It's just little pieces of the genome which are being made. So you're amplifying specific pieces of the genome. And that's how, again, you can have this massive genome. And again, towards the end of today, we'll talk more about those genomes, those individual pieces that you need many, many copies of. And so that's the process, is that transcription. Again, coming from DNA, the way that RNA is made is practically identical to the way that DNA is made. And again, this probably comes from an evolutionary aspect. RNA probably actually came first in terms of the genetic material, and DNA was a later invention. I think viruses did it, but again, we can talk about that later in my virology course. Uracil and ribose. We had to mention here that RNA is usually single-stranded. I don't like that term very much, which is partly why I always put it in quotes, because RNA in and of itself may be a single strand, i.e. it's just got one end here and one end there, but it's almost always base paired and complementary in almost exactly the same way as DNA. It's just usually one molecule rather than two molecules that are then base paired with each other. And so most of the time, RNAs don't look like we had back here on this previous slide, like this. They look much more like this. They're really base paired, and how much single-stranded RNA do you see down here? Practically none. And that's probably true for most RNAs, even the messenger RNAs, which are just being used for being moved from the DNA going off to the ribosome. So very often it's the structure of that RNA. And anyone who's had biochemistry, structure basically means function. Yeah, your question.
We talked about reverse transcriptase.
Yeah.
But does the RNA ever replicate itself? Or is there ever a time when RNA replicates RNA?
So the question here is, does RNA then lead to RNA replication? In cells, no. In viruses, yes. Okay. At least as far as we know of. There may be some cellular exceptions. Again, this is biology. There's always exceptions. But as far as we're concerned for this class, no. RNA doesn't lead to RNA itself. Again, that's not true for viruses. But for cellular life, as far as we know it, that's the case. Once you've made RNA has all these really cool, important active roles, but very often that RNA, as you mentioned, is being used now to make proteins. That's through the process of translation. That's done by this wonderful molecular machine down here, the ribosome, which again, as I mentioned before, we talked about that tree or bush of life, completely conserved. All cellular organisms have a ribosome that looks a lot like this, and the sequences of the nucleic acid and the proteins that go into that are very, very similar between all different cellular organisms that we know of. Genetic code, in terms of taking that RNA sequence and converting it into a protein sequence, is almost universal. And we'll talk about how some of those differences probably came about. But the main thing here is really that translation is all about the RNA. You can actually get translation from a ribosome in the absence of proteins. So the ribosome is made up of a bunch of RNAs and proteins, but it's really all about the RNA. And again, 3 nucleotides, 1 amino acid. And then we'll talk a lot more about these transfer RNAs and ribosomal RNAs as we move along. So you've done that. You make the protein. Here's sort of a classic protein here. Proteins do all kinds of different things inside the cell. From a biochemist point of view, it's all about the enzymes, which have these really elaborate structures, but are mostly involved in making a relatively small, very specific binding site for usually smaller substrates which will be used to undergo enzymatic activity, so stimulate chemical reactions. So this example here, lysozyme, which chews up polysaccharides, of course originally found in viruses, makes it terribly splendid and worthwhile and interesting. So final overview in terms of thinking about molecular biology is this is how you could shrink all of those mice down to. You could shrink a bacterium down to this. You can shrink an archaea down to this. We're really going to be talking about how individual nucleotides get made into polynucleotides. So individual nucleotides into DNA and into RNA. Then what these things do. They can either lead to the production of proteins, which is talked about the ribosome, that translational process, but are also really important for making more of the nucleotides. It's that genetic information which is getting passed along. Proteins have catalytic function. Some RNAs have catalytic function, which are involved in making more proteins, but also involved in making more nucleotides. So this is sort of the diagram here of what molecular biology ends up looking like. Okay, any questions on this part so far before we go and talk about genomes? This is Dr. Singer's idea of genomes here. So what is a genome? There are, in fact, lots of different definitions of genomes. We're basically going to use these two. All of the genetic material of an organism, and that could be all of the extra nucleotides, nucleic acids, etc. But usually when people talk about genomes, it's this nucleic acid sequence. And so this is a nice example here of E. coli, scanning electron micrograph of that here, and then the about 5 million base pairs of DNA which contribute to that whole genome. Yeah?
All meaning mitochondria?
Yeah, they'll also will include mitochondria for those organisms that have mitochondria. Of course, bacteria do not have them. Yeah?
And plasmids?
This would also include the various plasmids, any kind of extra chromosomal DNA as well. So again, you can argue about it's the organelle genome, etc. But usually, and again, particularly for this course, we'll talk about it as being all of that. Plasmids, organelles, etc. That's all going to be part of the genome. And so I'd say the complete nucleic acid sequence of any given organism. And we've got, actually I went back and looked just a couple of days ago. I think there are about 10,000 whole genome sequences that are out there right now. So it's a pretty impressive number that we have. So what do genomes look like? One of the big surprises when people sequence these genomes is that where we are, which is up here, we've got a relatively big genome, but it's actually pretty tiny compared to some of these other organisms which we usually don't think of as being more advanced. So one of my favorites here is the amoeba. To look at this scale here, it's a log scale. So this is over 10 to the 11th base pairs in the genome of some of these amoeba, as opposed to the couple of billion that we have. A lot of plants have larger genomes. A number of amphibians have larger genomes. So larger genome doesn't necessarily reflect what we like to think of in terms of complexity. A couple of big differences here. So it's not really so much the size that matters, has much more to do with how much of the coding sequence that you have there. So if you look at bacteria, and particularly archaea, down here at the bottom, these have relatively small genomes, but they're packed. Almost all of the genomes for bacteria and archaea are protein coding. If you look at E. coli, 11% non-coding. Some of these archaea here actually have about 2% non-coding sequence. And when I say non-coding, that's not coding for protein or for some kind of functional RNA that we know is a functional RNA. On the other hand, Humans, 1.5% of our genome is coding for protein. There may be a lot more that's coding for functional RNAs, and that's a really open area of research right now. If you look at the amoebal genomes, most of those seem to be not coding for protein at all. Lots of junk DNA, but we'll get back and talk more about junk DNA as we move on through the rest of the class. So, how do you get these big differences in the sizes of genomes? They're basically sort of 4 different ways that can happen. But probably most important for us in terms of thinking about different sizes of genomes, they're going to be from either gene duplication, where you go from 1 copy of your gene to 2 copies of the gene, or what's called horizontal transfer, or lateral gene transfer. Now, so what lateral gene transfer means is that you get a gene from one organism that shows up in another organism. Now, classic example that we talk about in terms of horizontal transfer would be sex. 2 different organisms exchange genes. That's what happens. You're not necessarily getting more there, but you're certainly getting a lot more diversity in that process. Also, viruses are great at transporting different nucleic acids around. In fact, they're probably one of my favorite cases there. But once you've done this gene duplication or horizontal transfer, then you need to generate the diversity. So all of these cellular organisms probably started at one particular place. We saw in that tree right at the beginning. There was what's called the last universal cellular ancestor, or LUCA, nice name, that probably had a relatively small number of genes, which have now diversified to give us this really amazing diversity of cellular life that we have right now. How did that happen? Point mutations, probably pretty few and far between, but over billions of years of evolution, there have been enough of these. Probably much more important are these moving around of genes inside of genes, so different pieces of genes that have been mixed and matched, also known as recombination. And we'll talk much more about the process of recombination, how that happens, but we know, certainly from looking at sequences, this is an extremely common event that takes place. And that brings me to a little bit of terminology. Unfortunately, this is something which is used usually incorrectly in the literature. Has to do with homology. So homology, by definition, means that you have common ancestry. Homology does not mean that you have a similar sequence. You could have a similar sequence, which is an indication of homology. But just sequence similarity does not mean that these things are necessarily homologs. They may or may not have come from a common ancestor. And you can certainly have things which have come from a common ancestor that don't have any sequence similarity relative to each other, because that sequence similarity over the billions of years of evolution has completely disappeared. You can get homologs in 2 different ways, either through speciation. So you have one organism here, say that LUCA that was at the origins of all cellular life, diverged to give different organisms. In each of those different organisms, that gene is changed. And so that gives you so-called orthologs, because they're in different species. On the other hand, you have gene duplication, which can happen in one organism. We just talked about that process, and again, we'll talk about how that happens more later on. And then 2 genes there, which then are going to be similar to each other because they have this common ancestor. They're going to be homologs. And these are paralogs because they're present in the same organism. And it's this process right here, paralogs, which really generates a lot of the diversity that we see in our genome sequences. And that's particularly true for gene families. And we'll talk a lot about gene families again as we move through the rest of the class. These are going to be different. Here we've got the red set of organisms, the blue set of organisms. These are now, and back up here, the gene duplication happened here. We had our tubulin gene. duplicated. We have alpha tubulin and beta tubulin. Now we've got two different species which have then arisen, each of which has these alpha tubulins and beta tubulins. So if you look at the alpha tubulin in a fruit fly here, it's also going to have a beta tubulin. These are now paralogs of each other because they're in the same organism. The alpha tubulins, which have now become different relative to each other, and again, this is a tree just like we had right at the beginning. Different sequences, different lengths of branches are how related they are to each other. The human alpha tubulin and the fly alpha tubulin are more similar to each other than the fly alpha tubulin and the worm alpha tubulin, but all of these are more similar to each other than their paralogs that are the same organism but a related gene. All of this would be a gene family. You've got the alpha tubulin gene family, the beta tubulin gene family. So everything here at the top, these are our orthologs because they've come from this speciation event, each of those individual different ones. And the paralogs are all the ones which are present in one organism, two different genes. Does this make sense? Some of this is rather confusing to people. Yeah?
It was something you said at the beginning about homology that's a little confusing because you said it's not necessarily determined by the gene.
Okay, it's not necessarily, so the question here, and I'm going to repeat your questions partly because I'm recording too, so, but the question is, homology is not related to the gene, and maybe I misspoke as far as that's concerned. So homology is not necessarily a sequence similarity. And so a homolog just means it's from a common ancestor.
So how do you determine that if it's not based on genetic sequence?
We'll get back to that. So the question is, how do you determine if it's a homolog if you don't see any sequence similarity? Turns out it has to do with structure. So we'll get back, but we'll get back to that when we talk about proteins on Wednesday. So it's a great lead-in. But yeah, very often it's actually hard to tell what a homolog is, and particularly if it's a homolog which has now had so much sequence divergence that there's not that much sequence similarity to it. It's a great question. And then your comment down here in the front about how do you get lateral gene transfer. Sex is certainly one way to do that, but viruses, this is what they do. They move genes around. And so that's certainly another way that you can have lateral gene transfer take place. So now I've talked about some of the mechanisms. Let's talk about a couple of the genomes in the last 10 minutes of class or so. Our textbook, it says you need fewer than 500 genes. It's kind of out of date because there was a publication in Science in 2006 with an organism that had 182 open reading frames. That open reading frames, so that's what's coding for protein, which is really pretty amazing. That's Carcinella. It's down here. This is the GC concentration, which is not as critical, but just the size of the genome in terms of megabase pairs. Megabase pairs is just millions of bases. We'd be out here, gigabase pairs. These are just bacterial genome sequences. And Carcinella is really way down here. Tiny, tiny, tiny little genome. Turns out that these are parasites, and so they've actually lost a lot of their genome, and you can tell that by doing some sequence comparisons, etc. What do they still hang on to? What makes it a cell? Translation. So translation seems to be something that you really absolutely have to have. It's something which is maintained in all of these different cells, even those which are very much a, excuse me, these are parasites. If you look at some of the bacterial genomes, I think they mention in the text, I can't remember exactly, that usually they're around 5. Some are actually up to about 10 million base pairs in size. Some of the smaller eukaryotic genomes actually overlap with that as well. And just as a quick reminder, again, people in microbiology know this already, there's more than 100 times more genes in our gut microbes than there are genes in the human genome. So our gut's way more complicated genetically than we are. And that's, in fact, a really big area of research right now, is to trying to figure out what those genes are and particularly what those genes are doing. So of those, again, I mentioned these smaller genomes, everything that's really conserved and everything else. Lots of these are protein families that have been generated through gene duplication, paralogs and orthologs relative to each other. Really, really common. The things that you find in basically every cellular organism are going to be these genes that are important for translation, for transcription, and replication. So again, these things that are really fundamental in molecular biology. It's making your proteins, making more DNA, and then making the RNA in that process. So these are things which are really, really well conserved relative to each other. That brings me now to model organisms. We'll finish up the lecture today on this, which is getting back again to that whole tree that we talked about in the first place. If you want to understand the fundamentals of how everything works, and that's basically what molecular biology is about, and it's best to study really diverse organisms and try and find those common threads that are really linking these things to each other. And of those things, bacteria, archaea, and eukaryotes, not surprisingly, makes a lot of sense. If you want to understand these real fundamentals, Abacterium and archaea, okay, it's not going to tell you everything about that, but it's the commonalities which are really important here. So for bacteria, E. coli is really the major organism. Most of the genomes of E. coli are about 4.5 million base pairs in length, relative to our human genome at billions of base pairs in length, 3 billion for our haploid genome, 6 for diploid. My favorite organism, Sulfolobus solfataricus, 3 million base pairs, about 3,000 genes in it. So those are great in terms of understanding, again, the fundamentals of molecular biology. If we want to understand something about us, not surprisingly, we're anthropocentric, we're interested in us, you might want to look at eukaryotes. If you're interested in things that are common across all eukaryotes, you're going to want to look at a diversity of different eukaryotes. That brings us to yeast, Saccharomyces cerevisiae, C. elegans. We'll take some pictures of this in just a second. This is the roundworm. Actually, Dr. Estes here in the biology department works on C. elegans. Arabidopsis thaliana is the classic plant. It's actually got a relatively small genome for a plant, only 142 million bases. Drosophila, the fruit fly. Anyone who lives in Portland, Compost, know all about fruit flies. Get lots and lots of them. And then mice as being sort of the closest relative to us as far as a lot of how our development works. These are what these guys look like. Again, yeast up here doesn't look very much like us, but in terms of its molecular mechanisms, you remember that tree, fungi are extremely similar to us, again, at a molecular level. C. elegans and Drosophila. Some of the main reasons that people use C. elegans and Drosophila is there's really good genetic tools for working with these, and we'll talk much more about genetics as we move on through the course. Basically, I like to think of geneticists who break things. So a geneticist will break something and then try and figure out what they broke. Turns out it's really useful in, again, both C. elegans and in fruit flies. For development, Xenopus is extremely useful, as is zebrafish, and we've also got a couple of people working on zebrafish here in the biology department. And then finally, the mouse system, and this is just a nice example here of one particular gene mutation that you have in mice and in humans. That particular gene mutation gives you basically an identical phenotype. It's a tyrosine kinase receptor that, again, we'll be talking about much more in the cell biology part of the class. Just finally here, another way of thinking about how common things are in terms of molecular biology is also to look at the history of the divergence of these different organisms from each other. And so if you look at how similar things like humans and chimpanzees are, it's only probably about the last 5 million years or so which these two different species have diverged from each other. whereas if you look at humans and some fish, it's probably hundreds of millions of years ago. Humans and bacteria and archaea, billions of years ago. And so again, if you're looking at these things that are going to be common across all kinds of different organisms, you're going to want to look at things that have diverged much longer ago and also have been maintained throughout evolution for billions of years. This is clearly something which has worked and continues to work to this day. So we'll look at systems biology. This is all you really need to know about systems biology. It's all about trying to understand the circuits which are going on. This is a really simple one. We'll talk about some of these more complicated ones later on in terms of the rest of the course. See you on Wednesday.